Okay, in this video we're going to talk about a third organic group of molecules, and that's proteins. Proteins are really important in your body. You can look at this picture and you see that there's meat and fish and cheese and milk, beans, all these nutrients you're supposed to eat, regularly to be strong and have strong muscles, right? So why do we need proteins? Well, proteins make up the following. First off, muscles. Your muscle fibers, they're specifically called actin and myosin. They are proteins. Enzymes, the enzymes that digest our food in our digestive tract are all enzymes. Enzymes govern chemical reactions. They allow chemical reactions to occur in our body much more rapidly. Antibodies. Antibodies are proteins that are sort of part of our defense system against bacteria and pathogens. So when we have an infection, antibodies will bind to and sometimes punch holes in pathogens or bacteria and help kill them. We also have white blood cells that are involved in our immune response, but antibodies are defensive proteins. And then structural components. Collagen is a protein. It's in our blood vessels. It's in our connective tissue. It helps keep us together, helps keep bones attached to bone. And then keratin's in our hair, keratin's in horns and hoofs and in your fingernails. So these are structural components, right? They help protect us. And then lastly, proteins are involved in what I'm just calling transport components. So hemoglobin, delivering oxygen around your body. Hormones, we talked about steroid hormones, specifically sex hormones, which are lipids, but there are protein hormones. An example is insulin. And then hormone receptors. These hormones bind to proteins on our cells, and that initiates a response within those cells. So the hormone has something to bind to, and that is a protein called a hormone receptor to initiate that response. So these are some of the functions of proteins. Again, looking at proteins, we want to then start with the basic unit. The basic unit of a protein is called an amino acid. And so the monomer is an amino acid. Amino is named for this group right here, NH2. That is an amino group. And then this carboxylic group is actually a weak acid. It's not a really strong acid, but it is an acid. So you put them together and you have an amino acid. And then the third part is this R group up here. Now R is not an element on the periodic table. R is just an abbreviation for one of many functional groups. So for example, leucine is an amino acid. We have 20 different amino acids in our body, but leucine, the R group, is this complex right here. As you can see, a bunch of carbons and hydrogens. Now this is going to be nonpolar, right? That's going to mean that leucine is going to want to turn away from water, that R group. Whereas if we look at serine, serine has that hydroxyl group on it. And just like sugars and other polar molecules, that's going to be attracted to water. So not all amino acids have the same properties. Some are hydrophobic, some are hydrophilic, and this is important when we get to the structure of proteins, because if we have a mistake in that sequence, we're going to have proteins that have very different properties. Okay, so if we connect a bunch of amino acids together, we form this polypeptide. So the polymer form of a protein is called a polypeptide, named actually for the peptide bonds that hold them together. So amino acids are the monomers. They're connected together to make polypeptides. Polypeptide is also synonymous with a protein. There's a slight difference, but for biology, we're just going to treat them as one and the same. Then these proteins have to get folded up into a specific shape. So if it's a globular protein, it might look like this one. This is an enzyme called lysozyme. It breaks things up, but it's got to have that specific shape. If it's a keratin in your hair, it's going to be a long, straight protein, or if you have curly hair, it might be a curlier protein with more kind of curves in it. So each protein has a specific shape, and that shape is based on its function. So an important understanding of proteins is that they need to have that specific shape, and they can be denatured. If you denature a protein, you change its shape physically. Okay, so we have maybe the normal protein over here on the left, and then maybe we've heated it up and we've caused it to change its shape. Okay, now it's denatured. And you've seen denatured proteins. In fact, you've probably denatured proteins if you've fried an egg, right? You drop that egg white is pure protein. Drop it on a hot frying pan, it turns white. It's getting denatured. It goes from liquid, clear, to solid white. That's a denatured protein. Another example of a denatured protein is sour milk. This is actually someone making cheese, and to make cheese you take milk and you put acid in it. The acid denatures the milk into curds and whey, and then you take the curds and you dry them out and you process them into cheese. But basically you've got to start by taking milk, a liquid, and denaturing it and turning it into a solid in the first step of making cheese or yogurt. And then another example, this over here is called ceviche. Now, if you're not into Spanish or Portuguese food, you may have never had ceviche. Ceviche is just fish, and if you look at it, it looks like normal cooked fish. But the interesting thing about it is this fish is cooked without any heat. It's cooked with lime juice. So they basically take raw fish, soak it in lime juice. Lime juice is very acidic, so that acid actually denatures and cooks the fish. And then they serve it cold. So when you're eating ceviche, you're eating basically cooked, denatured fish that's actually not been cooked. So there you go. Try it next time you're at a Spanish or a Portuguese restaurant. Ceviche. It's very good. Okay, so an important function of proteins, or an important group of proteins that we're going to talk about, are enzymes. So we're going to do a big enzyme lab this week. So enzymes are proteins that speed up chemical reactions in the body. So I talked about digestive enzymes. So in your intestines, you've got an enzyme called sucrase, and maybe it looks like this blue structure. And it grabs onto sucrose, a disaccharide, and rips it into two monosaccharides so that your body can then process those. So that's an example of an enzyme, and the substrate is basically what it's acting on. So in this case, if the enzyme is sucrase, it's acting on the disaccharide sucrose. So the sucrose is the substrate, sucrase is the enzyme. Enzymes typically end in ASE. It's an easy way. If you see something ending in ASE, it's an enzyme. So how do enzymes speed up chemical reactions? Well, they do it by lowering the amount of activation energy. The activation energy is the energy that's needed to kind of get the reaction going. It's kind of like pushing that rock to get it to fall off the cliff, or the spark in your engine that's needed to start your engine, to start that gasoline burning. Okay, and so this diagram, this is kind of a scientific diagram, and it shows the normal amount of energy needed to start a reaction. Let's say you've got sugar and you want sugar to burn. You've got to heat that sugar up to about 450 degrees Fahrenheit. If you have enzymes, which you do in your body, you can burn that sugar at body temperature at 98 degrees Fahrenheit. So that lowers that activation energy. Okay, and then it produces obviously lower energy products when you burn that sugar. An easier way to think about that might just be thinking about this activation energy is kind of a barrier to get over. And if you lower that barrier, you knock some of the rocks off of that fence or that barrier, it's just easier for those frogs to jump from one pond to the other. So essentially all we're doing is lowering that barrier. So let's look at how that happens. So here's sucrose, the disaccharide, and it's going to be chopped in half, what we call a a hydrolysis reaction, because we're putting water in, not taking water out, a hydrolysis reaction. And it's going to produce two monosaccharides, glucose and fructose. Okay. Well, you don't see the enzyme in here. The enzyme is not a reactant on the left or a product on the right. But if we look at what happens and what the enzyme is doing, you can see here that here's your sucrose, and it's going to bind in step one to the enzyme. And enzymes are really like clamps, in that they just kind of hold that substrate in a way that that bond can be broken or a bond can be added very easily. So kind of like if I'm trying to saw something and I can't really get a good grip, if I put it in a good clamp, it's going to be easier to break it in two. Think about it like that. So that enzyme kind of holds it in a way that that bond can be broken, and that hydrogen and the water molecule can be split and inserted in there, and there's the OH and the H that are stuck onto glucose and fructose. And then those two monosaccharides can be taken into our blood and delivered to cells and used for energy. Okay. Now the enzyme is not created or destroyed, it can continue to break down more and more sucrose. So unlike reactants in products, the enzyme governs the reaction and can be used again and again. Now, the last thing about enzyme activity is that there are environmental factors that can affect them. We talked about how temperature and pH specifically can denature proteins. So for a human enzyme, if it's too cold, there's not enough kinetic energy. Temperature measures the amount of kinetic energy in molecules. So if molecules are moving very slow, enzymes are going to go very slowly. So cold-blooded organisms, if they're not warm, they're not going to be able to move very fast. Their muscles aren't going to be able to contract. So a lizard or a snake in a cold morning is going to try to find a nice warm rock to sun on to get warm quickly. So those enzymes work quickly. We're warm-blooded, so we keep our temperature at the optimal rate for our enzymes. If you're a thermophilic or heat-loving bacteria, that might be close to the boiling temperature of water. But for us, we like a temperature right around 38 degrees Celsius, which is about 98 degrees Fahrenheit or body temperature. Once you get above that temperature, molecules are moving really fast and reactions can go really quickly. But again, high temperatures tend to denature proteins and enzymes. So the enzymes get denatured. Okay? Same thing with pH. You have actually a digestive enzyme in your stomach called pepsin. It likes a pH of about 2. Your stomach is very acidic. If the pH gets too high, then it doesn't maintain the right structure and pepsin can't work. If the pH gets too low, pH around 0, that's also going to denature the pepsin and it won't work. Then as that food goes into your intestines, there's another enzyme that breaks down protein called trypsin. It likes a pH of right around 8. So we need to, again, adjust the pH of the food from the stomach to the intestines so that the trypsin can work in that pH. So again, part of homeostasis is maintaining proper temperature and pH internally so that our enzymes can function. Because proteins, if they're not within a very narrow temperature or pH range, they will get denatured.